laboratory of David Mills at the University of California, Davis studies the microbial ecology of food and beverage production. Recent work in the Mills lab involves using next-generation sequencing to profile bacteria and fungi, including yeasts, in vineyards, wineries, breweries, and creameries. They discovered evidence supporting regionality, or terroir, of microbes in vineyards from wine-growing regions in Northern California.
So the Sloan Foundation Award will give us an ability to try to address some of these questions. The concept of microbial terroir as it's associated with food products that we've made some recent discoveries in begs the question, does that microbial terroir enter into the facilities and populate the facilities as well? So when the grapes are harvested and we know that there's a large amount of microbiota that comes into the facility, does that microbiota persist in the facility over time? Does it persist through all the various practices and changes that the building undertakes throughout a season? And in dairies, it's the same thing. We have multiple visits of milk to a dairy, of course, over a year, and there's a seasonality to the microbiota that comes into that facility. Yeast and fungi are important in both wine and cheese fermentations, both as spoilage and as beneficial microbes. A critical asset to this project is Dr. Kyria Boundy-Mills, who curates the Pfaff yeast culture collection and is providing expertise in fungal taxonomy.
One of the features that distinguishes this project in particular, looking at dairies and wineries, is the challenging condition of having an environmental space which is uncontrolled. Typically deliveries of product coming from grapes in vineyards into unconditioned spaces with lots of outside air, lots of movement by people and by equipment from room to room. The challenge for us now to extend the work on the microbiome into the building environment is to better understand the building environment. And we intend to do that in this project by measuring not only the temperature and humidity, but also things like the carbon dioxide content, which would tell us more about a fermentation environment. Volatile organic carbons, which are other than CO2. Trying to follow dust particles and trying to follow human traffic in and out of the room. By measuring these properties in the fermentation hall and in the adjacent rooms, we believe we can do the balances to help us understand the relative contributions of airflow from different rooms.